Good afternoon. This is Dr. Javeria Altaf. I'm working as an assistant professor in the Department of Zoology in Government College University, Faisalabad, Pakistan. Today I'm going to present my research journey from 2004 to 2024. So what was my vision? My vision was to assess the role of invasive species in the environmental bioremediation and uh, to control these invasive species through some biological method or some other strategy which is equally helpful for the human beings and in a way that does not involve any chemical method because that further degrades the ecosystem. And why we were using these resistant species for the environmental factors and for the to study, it was because they're resistant in nature. And first of all, why I wanted to work on these resistant species, because there are no ethical problems when you're working on them, and when you're publishing them, that there are no ethical issues. That was the basic initial ease which I got out of these. And they are generally resistant to any kind of change, environmental change. And due to this, they are very easy to culture in the lab, and we do not need much economic support to maintain the temperatures and all these things. So that is the reason I took these species. And later I also found that definitely these species which are potent enough to live in a very harsh environment or in sometimes in a very polluted environment, they have very good antimicrobial compounds in them. So that was one reason why I chose these species. So this is my brief introduction. And I chose, initially we started working on the channets. Channets is a very valuable fish and it is very tasty. And it's a freshwater fish. And mostly the origin of this fish has been reported from Africa. Uh, these were used in my initial studies to control the tilapia, which is a pest species. It's an omnivore. It competes with the local fish fauna of our country. And so there is a dire need to control these species. So that is my first research work when I was working in Masters of Zoology. And then we later barcoded these species. There are 4 species in Pakistan, and we barcoded all the 4. And now we are working on the supply chains and for the QR coding of these species, so that we can export it in the form of food through. If we do have a QR code, then definitely we can export our uh, fish uh, in a more reliable way. And then we are also working on barcoding of lumbricids. These are earthworms, and again the species of Lumbricidae, family Lumbricidae, which is Lumbricus terrestrius and Lumbricus rubana. They are again very, and we are using them for the vermicompost purpose. So that is again a product which we are getting out of it, and it is it's ongoing. One of my PhD student is working on this, and we are almost in our completion phase. During my MPhil and my PhD, I worked on the taxonomy and ecology of the gastropods. Gastropods are invertebrates. They belong to phylum Mollusca, and they're very small animals, but they are generally in here in Faisalabad in Pakistan. We consider them as pests, and when we find them in our fields, we generally spray them, and so that it does not enter into fodder for the cattle or into other crops. So we do not want uh, a loss in our yields. So that is the reason why we generally spray them out of our agricultural fields. And the sprays are very toxic, not only for the environment but also for the human physiology and animals as well. So in that, during my MPhil and PhD, I worked in the biodiversity and ecological studies of the gastropods, and there were various findings. Then, in the later stages, when one of my student was working on the gastropods, so we decided that we would use certain species which are really pest species again, and they're invasive in nature. They belong to genus Physa. They are mostly physids, and we use them to assess their antimicrobial potential. So this is my basic and brief introduction. This is the map of Pakistan in blue color. And then there's one region of Pakistan, which is Punjab, that is one province. And in the central Punjab, there is a city, a very large district, which is Faisalabad. So this was our study area throughout the research journey. And it's generally considered to be it is generally said that Faisalabad is the Manchester of Pakistan. So because it is a hotspot of all the industrial activities. This was the hypothesis of my PhD research work. The phenotypic similar populations of the gastropods have genotypic polymorphism, and we worked on the morphological markers of the species and as well as on the genetic markers of the species. Then the agroecosystem of Faisalabad has a high species diversity of the gastropods, and the gastropods have a strong interspecific relationship and strong relationship with host crops and environmental variables. The terrestrial gastropods are the bioindicators of the environmental degradation. There were 5 publications in my PhD research work, which were titled Taxonomic Studies on the Occurrence of the Snails in the Agroecosystem, Genetic Diversity Analysis of the Snail Species using Molecular Marker Technology, Assessment of Diversity and Distribution of Snails in the Agroecosystem of Faisalabad, Interaction of the Snail Species in the Agroecosystem of Faisalabad, and Terrestrial Snails as Bioindicators of Environmental Degradation. And these were the findings. 15 species are present in the agroecosystem of Faisalabad. Rapid markers assisted studies showed that physids were distantly related to the terrestrial snails, and the diversity indices was highly significant in the crop fields and non-significant in the ditches, which might be due to the polluted waters. There was overall a positive interaction among all the species. Moving from north to south of the Faisalabad, the species belonging to genus Physa, Zooticus, and Sesaloids were reducing, while others have achieved the status of a pest. Members of the family Phycidae and family Subulinidae were found resistant to the environmental degradation, and species of these families can be used for the ecological bioremediation. Based upon the findings of the previous studies, we have hypothesized that multiple species belonging to genus Physa are present in this region, and the Physid species populations are highly polymorphic. So we published these 2 articles out of our studies, and on the left side we have found these are the Physid species, and we found 3 Physid species, Physa acuta, Physa fontanalis, and Physa gyrina. And similarly, in another research work, we found 3 different species of genus Zooticus. This was Zooticus pullus, Zooticus insularis, and Zooticus Xion. And these 2 families, which belong to, these snails generally belong to the family Phycidae and family Subulinidae. So we further worked on these species. So physids were found from a number of villages of central Punjab due to its invasive nature, and the principal component analysis showed that the shell length and shell width were most prolific components for the identification when we are doing morphometry. The square distance and discriminant analysis showed that Physa acuta resembles with Physa fontanalis, and both these species do not resemble with Physa gyrina. Only 6 specimens of the Physa gyrina were found, and due to what we thought that there is a need for the further taxonomic reconsideration of the species. Physids are invasive in nature, and it was hypothesized that physids may bioaccumulate pollutants from the water and soil for its bioremediation, and because they are, as they are living in such a harsh environment where a lot of pollutants are being released and where a lot of agrochemicals are being sprayed on, and they are surviving in that condition. So possibly they do have a very strong resistance against various other stresses as well, such as microbes. So this was the hypothesis of our further studies. So we had hypothesized that physids are very important species for the bioaccumulation of various heavy metals and pollutants. So we planned a study in which we used 4 heavy metals that are highly carcinogenic, and they are in group 1 highly carcinogenic heavy metals, that is lead, arsenic, cadmium and chromium. So we sampled 62 different villages from where we took samples of soil and water and our gastropods, and we analyzed the sample, we processed the sample, we calculated the concentration of heavy metals in the soil, water, shell and body. So, and then we use certain predictive models to assess that how much risk is there of the human cancer in the region. And out of that, we published this article. The maximum permissible limit for the water quality index is 100. And here you can see that water quality index is much higher than 100 in all the in all the clusters of villages. We took 62 villages for the sampling purpose, and we then divided them into 15 different clusters. So one cluster has more than one village. This way we carried out our sampling, and but when we found the bioaccumulation factor, it was found that in the 3 species there was no difference. There was no significant difference in the 3 species of physids for the bioaccumulation of the heavy metals. Now you can see that the HQ of water through oral ingestion is much higher than 1, which shows that how hazardous water is for the human consumption. And the cancer risk factor shows that the cancer risk is very high in the region, especially in RB-R5 cluster of villages, where 1 out of every 10 individuals, adults, are at the risk of cancer. Similar results were found in case of the children, and rather more drastic results were found in the same cluster of the villages for the children, with HQ much higher than 1 for the water ingestion. And here you would find that there are approximately 19 children out of every 100 children are at the risk of the cancer. Since we had identified certain invasive genera in our previous studies, so we tried to compare them in terms of the antimicrobial activity. So we did a comparative evaluation of the antimicrobial activity of the foot mussel extracts from the genus Physa and genus Sesiloides. Out of 3 species of physids, we used 2 species of physids because Physa gyrina was found in very less numbers, as we have previously discussed in the previous slides. So we just took 2 species, Physa acuta and Pfizer fontinalis, and we prepared the body extracts in 3 different solvents, that was ethanol, acetone and diethyl ether, and we coded them as SS1, SS2 and SS3 and SS4, SS5 and SS6. So then we checked their activity against various microbes, and we used an antibiotic as a control. The results were impressive, since it was not a purified compound. It was just a crude body extract, and it was giving good results in that terms. Similarly, we did an antifungal assay, and the results were uh, similar in case of SS4. For the antiviral assay, we used heme agglutination test for against Newcastle virus, and the results were good and impressive in this case as well. But when we carried out anti-cancer assay using kidney cell cancer cell lines and liver cancer cell lines, we found very contrasting results. In case of control, you can see that there is 100% proliferation of the cell lines. But in case of our 6 extracts, the proliferation was even more than 100. So due to what, we did not proceed it further to identify that what particular factors are there or what particular compounds are there which can act as anti-cancer, because the results were showing that it was proliferating the cancer. However, it was uh, in uh, in agreement with the previous work. I have shown that human cancer risk factor was very high and in the region. So now you can see in the assays as well that the proliferation was very good and very high um, in case of the due to these body extracts. It means that they are by accumulating all these pollutants that are cancer causing. Then we characterize the compounds of these present in the body extracts using ESI-MS. So we identified the compounds through GNPS, and we identified approximately 162 compounds, and 67 have not been reported for any activity, and 76 compounds have been reported for the antimicrobial activity, and 23 have been reported for the anti-cancer activity, and there were 13 which were reported for the anti-inflammatory activity. So a number of compounds are there, and further analysis will lead us to even more compounds. Uh, but up till now, this is our these are our findings. But the point is that I've not yet shared the list of the compounds which I have with me. I haven't shared it because since I have shared all the details of my species, so I think that uh, it was not legible to share it right now, as I have not published the list right now. So, however, I have shared my species from which we have obtained these chemicals and compounds. Just for the sake of interest, I have shared this slide that we have developed a product from Lumbricides, and we did the morphometry of Lumbricide earthworms, we did barcoding and QR coding of these species, and now we have developed a vermicompost. And soon we will be doing our work on supply chain development as well as QR coding, so and patent registration. So we are yet working on that as well. Soon we will be sharing our data with respect with of these findings with the audience. Thank you so much. This is all about my presentation, and this is all about my research journey. And if you do have some questions or you have some suggestions, then I would welcome you